Imagine this, a biological countdown clock, and it just starts ticking the exact moment a seemingly normal fever hits you.
Yeah, and it's a clock you can't hear.
Right. You don't hear it, you definitely can't see it, but within about 24 hours, your brain's internal environment is just violently overthrown. It's, well, it's honestly one of the most aggressive and fast-acting medical emergencies on the planet.
It really is. By the time the obvious symptoms actually show up, you're already losing the fight.
Exactly, which is why we're so glad you're joining us for this custom-tailored deep dive today. We're getting into a really paradigm-shifting piece of research. It was published by Karger in the journal Neuroimmunomodulation.
Right. The paper is titled Challenges in the Determination of Early Predictors of Cerebral Malaria.
And look, we are completely skipping the textbook definitions of mosquito vectors today because, well, you already know how this disease spreads. We're plunging straight into the bleeding edge of diagnostic prediction.
Which is so needed.
Yeah.
Because malaria, I mean, it still claims over 2 million lives a year globally.
Wow, 2 million.
Yeah. And the specific complication we're focusing on, cerebral malaria, it primarily targets children in sub-Saharan Africa. And it operates on this incredibly unforgiving timeline. The neurological damage just outpaces our fastest treatments.
So our mission today is to figure out the molecular battlefield inside the brain. We're looking at the fatal flaws in how we currently diagnose comas in endemic areas and how observing the tiny microscopic motor changes in infected mice might actually allow us to build a mathematical early warning system for humans.
It's a huge fundamental shift in strategy. For decades, medicine has essentially been reacting to the crash.
Right, trying to fix it after it happens.
Exactly. But this research, it's about finding the mathematical signal to predict the crash before it ever happens.
Okay, let's unpack this, because before we can really get to the prediction part, we have to look at the absolute diagnostic nightmare that doctors are facing on the ground right now.
It is a nightmare. I mean, there are 5 species of Plasmodium that infect humans.
Right, there's Vivax, malaria, oval, Nolici, and of course, falciparum.
Right, falciparum is the heavy hitter here. It's the parasite strain that drives the vast majority of severe neurological complications.
But diagnosing cerebral malaria, or CM, in a busy rural clinic, it's not like, looking for a broken bone on an X-ray. It's incredibly murky.
It's so murky. The World Health Organization specifically defines cerebral malaria as a coma persisting for at least 1 hour after a seizure or after correcting blood sugar.
Okay, so 1 hour.
Yeah, but the standard protocol on the ground just assumes that if a patient, especially a child, arrives at a clinic in a malarial zone in an unexplained coma, well, you just treat it as cerebral malaria.
Because it feels like the safest bet, right?
It does. But the data from a major study in Tanzania completely shatters that assumption. It's kind of wild.
Oh, wow. What did they find?
Well, they looked at patients who were admitted with a clinical diagnosis of cerebral malaria, and they found that only 19.7% of them actually had detectable parasitemia in their bloodstream.
Oh, wait, less than 20%.
Less than 20%.
That means 4 out of 5 patients in that study were basically misdiagnosed. I mean, that's like playing a high-stakes game of medical whack-a-mole.
That is exactly what it's like.
Every single severe neurological symptom gets slapped with the exact same label just because they happen to be in an endemic zone. It's hospital triage running blindfolded.
And the downstream effects of that blindfolded triage are just catastrophic. Because when clinicians aggressively treat every unexplained coma with antimalarials, they stop investigating.
Oh, I see. So they stop looking for what's actually causing the coma.
Right. The underlying pathology gets completely ignored. And that could be viral encephalitis, it could be a bacterial brain abscess, or even heat stroke, or an acute metabolic crisis.
So the patient doesn't die from malaria, they die because their actual condition was just masked by the assumption of malaria.
Precisely. And beyond the immediate danger to that one specific patient, carpet bombing every single fever and coma with antimalarial drugs, it puts immense evolutionary pressure on the parasite itself.
Because we're basically training the parasite to survive our best medications, right?
Exactly. We're accelerating the global crisis of drug-resistant strains. We're wasting our best weapons on shadows, basically, while the real targets evade us.
Wow. Okay, so identifying CM is like finding a needle in a haystack of other diseases.
Right.
But to make matters even worse, from what I read in the text, the disease literally shapeshifts depending on the age of the patient.
It does. It's wild how different it looks. The clinical profile in a pediatric ward looks absolutely nothing like the profile in an adult ICU.
Well, let's focus on the children first, since sadly they bear the absolute brunt of the mortality rate. How does it usually start for them?
Well, the presentation usually kicks off with about 1 to 4 days of fever, maybe some vomiting and a cough. But then the neurological cascade hits, and it hits fast.
Yeah, the data says about 60% of these children will suffer violent seizures before slipping into a coma.
Right. And they develop these very specific, unique visual markers. Malarial retinopathy is a crucial diagnostic footprint here.
Retinopathy, so changes in the eyes.
Yeah, if you examine the retinas of these pediatric patients, you see distinct abnormalities. Over 60% of kids exhibit vessel whitening.
Which means the capillaries in the eye literally lose their healthy color.
Exactly. You also see what we call cotton wool spots, which are essentially these microinfarctions. They're tiny areas of dead tissue in the nerve fiber layer of the retina caused by restricted blood flow.
And there are also those Roth's spot-like hemorrhages, right?
Yes, exactly. It's a visible proxy for the ischemia, the lack of blood flow happening deeper inside the brain. And tragically, most of those pediatric fatalities occur within the first 24 hours of admission.
That is just brutal. But then when you switch focus to adults who get CM, it shifts entirely.
It really does. In adults, the neurological symptoms are almost secondary. What you see is more of a massive multi-system organ collapse.
Right. The sources mentioned acute renal failure, severe jaundice from liver dysfunction, and pulmonary edema.
Yeah, which is fluid filling the lungs. And those distinct retinal spots we see in children, they are super rare in adults, only appearing in about 15% of adult cases.
And this adult presentation is geographically distinct too, isn't it?
Very. We see it far more often in low transmission areas like Southeast Asia or Papua New Guinea,
Right.
rather than the high transmission zones of sub-Saharan Africa.
Okay, I want to push back on that geographic division for a second, because if a child in Tanzania and an adult in Thailand are infected by the exact same falciparum parasite, but their bodies react with completely different syndromes, are we just looking at a biological age factor?
It's a great question.
Like, does a developing pediatric brain physically react differently to the parasite than a mature adult brain, or is it something else? Because the science still isn't totally sure, right?
Well, if we connect this to the bigger picture, this age discrepancy really ties into global immunity patterns.
Okay, how so?
In hyperendemic regions like sub-Saharan Africa, a human is bitten by infected mosquitoes continuously from birth. So their immune system is constantly sparring with the parasite.
Oh, I get it. So the children who survive those early years are essentially building a localized tolerance.
Exactly. They develop a level of clinical immunity. So by the time they reach adulthood, their immune systems have learned how to manage the parasitic load without triggering a totally catastrophic response.
That makes sense. That limits severe adult cases in much of Africa.
Right. But in Southeast Asia, transmission is often lower. An adult might encounter the parasite for the very first time at age 30. Their immune system is naive to it.
So when the infection hits, the adult body just panics and triggers that multi-organ failure.
Precisely. It's the historical resume of the immune system that dictates a lot of this.
Okay, so we know what it looks like on the outside and how age and location play a role. But what is actually happening inside the brain's microenvironment to cause this rapid decline?
This is one of the most intense, decades-long debates in parasitology.
Right. There are two competing theories of CM pathophysiology, basically trying to explain the mechanics of the crash.
Yeah, you have the sequestration hypothesis versus the inflammation hypothesis.
Okay, let's start with sequestration, which was first proposed way back in 1894. As I understand it, we should view this as a plumbing problem.
A plumbing problem is a great analogy. The mechanism here relies on the parasite hijacking the physical structure of the red blood cell.
Right. The parasite forces the infected red blood cell to express these specific adhesion proteins on its outer membrane.
Yes, PFEMP1 proteins. They essentially act like molecular Velcro.
So the parasitized red blood cells, the PRBCs, they become rigid and intensely sticky.
Exactly. And as they circulate up into the brain, they snag on the walls of the tiny brain capillaries. More cells pile up behind them, sequestering in these tight spaces and physically barricading the blood flow.
Clogging the pipes.
Clogging the pipes. The brain tissue gets starved of oxygen and the neurons suffocate. And postmortem autopsies do consistently show capillaries packed tightly with these parasitized cells.
It's a very clear physical mechanism. But then there's the inflammation hypothesis, proposed in 1948. If sequestration is a plumbing problem, inflammation sounds more like an electrical fire frying the brain's delicate environment.
That's a perfect way to put it. The inflammation theory argues that the physical blockage isn't the primary cause. It says that when the parasite releases its toxins, it triggers the host's immune system to absolutely overreact.
And that unleashes a cytokine storm.
Right. And a big part of that is what happens when a red blood cell is ruptured by the parasite. It releases free heme into the bloodstream.
Heme, like the iron compound.
Exactly. Inside a healthy cell, it's essential for carrying oxygen. But when it's floating freely in the blood, free heme is highly toxic. It creates severe oxidative stress.
So it's basically tearing through the tissue. And how does that affect the blood-brain barrier?
Well, the blood-brain barrier is maintained by astrocytes, these specialized lineal cells. When that cytokine storm and the toxic free heme hit, the astrocytes get damaged.
They get fried.
Yeah. The barrier becomes highly permeable. Fluid rushes into the brain cavity, causing severe brain swelling or cerebral edema.
And since the skull is rigid, that swelling physically crushes the brainstem, letting toxic malaria antigens flood the environment.
Exactly, leading to coma and death.
So it's likely a deadly combination of both, right? The clogged pipes and the electrical fire. But wait, there's a critical contradiction in the text here.
Yes, there is.
If it's purely a mechanical plumbing problem requiring those sticky proteins to clog the capillaries, how are we seeing increasing cases of cerebral malaria caused by Plasmodium vivax?
Right, because vivax is a parasite strain that doesn't sequester.
It doesn't make the cell sticky at all.
Exactly. It rarely sequesters in the brain. Yet it still drives patients into fatal comas. This biological contradiction throws massive weight behind the inflammation hypothesis.
It proves the parasite doesn't even need to physically block the brain to destroy it. It just needs to provoke the immune system into a panic.
And because that inflammatory cascade happens so fast, usually killing within 24 hours, waiting for a patient to slip into a coma before diagnosing CM is a losing battle.
Which brings us to the need for a crystal ball. We need a way to predict the crash before it happens. And here's where it gets really interesting.
Yes, the experimental murine model.
Right.
Mice.
Right. Scientists use a susceptible mouse strain infected with Plasmodium berghei ANKA.
Though we should add a crucial caveat for the listener here. Unlike humans who get red blood cells stuck in their brains, these mice get white blood cells, or leukocytes, stuck.
Right. It's a slight biological deviation, but the end result, the breakdown of the blood-brain barrier, the brain swelling, the fatal coma, it maps incredibly well to humans.
It does. And the researchers wanted to see if they could predict the coma before the mice actually looked sick. So they used the SHIRPA protocol.
The SHIRPA protocol, which is a standardized behavioral assessment for mice, right?
Yes, it features 40 different observational tests. They aren't just looking to see if the mouse is tired. They're checking gait, muscle tone, reflexes, even autonomic functions like piloerection.
Piloerection, that's when their hair stands on end, right?
Exactly. So they tracked and scored the mice on days 4, 5, and 6 of the infection to build a mathematical prediction model.
And the results are wild. Because on day 4, the test was essentially useless, right?
Right. On day 4, the predictive accuracy, the ROC, was under 60%. The sick mice were moving and reacting, just like the ones who would eventually clear the infection.
So what does this all mean for day 5? Because things completely changed.
Day 5 was the sweet spot. The total score on day 5 successfully predicted which mice would develop cerebral malaria with an 89% positive predictive value.
Meaning, if the model flagged them, there was an 89% chance they'd crash.
Yes. But even more astonishing, the isolated motor score on day 5 had a 100% positive predictive value.
100%? Wait, really?
Really. 8 out of 8 mice flagged by just the motor tests went on to develop fatal CM.
What's fascinating here is the timeline, though.
Yes, what's fascinating here is that, to the naked eye, these mice only physically show severe neurological signs a few hours before death.
Right, they look totally fine on day 5.
Exactly. Yet this mathematical scoring of super subtle behavioral changes can predict their fate 36 hours in advance.
36 hours. That proves early detection is mathematically possible. We're talking about a disease that kills humans in 24 hours, so buying 36 hours of lead time is massive.
It's game-changing. By studying the minute motor changes in mice, researchers are building the foundation for a clinical scoring system that can screen human patients upon hospital admission.
Right, you could run a 2-minute reflex test on a child before taking a drop of blood, long before a deadly coma sets in.
Exactly. It's not just an academic exercise, it's a race against that 24-hour clock we talked about.
That brings it all back home. But before we wrap up, we have to talk about the aftermath. Because surviving CM isn't the end of the story.
No, it's not.
The source notes that 10 to 25% of children, and some adults too, who survive cerebral malaria suffer from long-lasting neurological and cognitive impairments.
Yeah, the inflammation leaves a lasting mark. We're talking about severe personality changes, language disorders, and clinical depression.
Which leaves us with a really provocative thought to ponder. If the physical malaria parasite can be completely cleared from the blood by medication,
Right, the infection itself is gone.
right, then what does the persistence of these severe psychiatric and cognitive changes tell us about how profound systemic inflammation might permanently rewire the human brain?
It's a haunting question. The circuitry itself is altered.
Yeah. Could surviving this microscopic parasite leave a physical footprint on human consciousness itself? It's definitely something to think about.
It really emphasizes why predicting this disease is so much better than just trying to survive it.
Absolutely. Well, that's all for today. Keep questioning the unseen mechanisms around you, and we'll catch you on the next deep dive.